Pharming, the genetic modification of plants and animals to produce medicinal products such as vaccines, hormones, and protein-based drugs. Herceptin, also known as trastuzumab, an antibody and drug to treat breast cancer. Antibody, a protein that binds to target antigens and signals the immune system to attack foreign substances. The immune system can be triggered to produce antibodies once antigens are introduced. Antigen, a foreign substance that produces an immune response. Transgene, a gene that has been transferred from one organism to another. Plasmid, a circular piece of DNA found in bacteria. Transformation, the incorporation of foreign DNA inside a bacterial host. Agrobacterium tumefaciens, bacteria commonly used in genetic modification that has the ability to insert a segment of its DNA into the genome of another organism. Surrogate, a host. Regulatory gene, a gene that is involved in controlling the expression of certain other genes. What if there was an efficient way to mass-produce life-saving drugs? Take Sarah, a woman diagnosed with breast cancer who can't afford expensive treatment drugs. Take Plantform, a company that specializes in farming and has the means to produce Herceptin quickly and in large volumes. Using genetically modified tobacco plants, Plantform can manufacture Herceptin in an efficient way that lowers prices and increases availability of the drug, enabling Sarah and many others to fight the cancer. Herceptin, scientifically known as trastuzumab, is an antibody that binds to antigen HER2, high levels of which is found in those with breast cancer. At Plantform, the gene for this antibody is inserted into tobacco plants, chosen because they can grow quickly and produce many seeds at a time. When the plant has matured enough, it is thrown into a blender and the precious drug is extracted from the slurry. By growing tobacco plants instead of batches of cell culture, manufacturing costs could be reduced tenfold. Herceptin is just one of many examples of how genetically modified pharmaceuticals dramatically improve the production line. Ebola and HIV antibodies are manufactured by tobacco plants as well. Atrin, an anticoagulant that is used by people with clotting disorders, has been obtained through transgenic goats that can each produce up to 211 gallons of atrin-laced milk a year. In more recent developments, scientists have harvested rusin and edible vaccines. Rusin, a drug to treat angioedema, can be harvested from transgenic rabbit milk. Edible vaccines are antigenic proteins in the edible parts of the plant. When consumed, the immune system detects the antigen and produces corresponding antibodies, just as how regular vaccines would work. So how is farming carried out? First, there are various methods to identify the gene for the protein of interest. The genome of an organism with the desired protein can be compared to a genome of the same species without the presence of that protein. Scientists may also delete parts of the genome until the desired trait is lost. In plants, the next step is typically transformation. Enzymes are used to cut and insert the gene of interest into a plasmid. The Agrobacterium tumefaciens is shocked with heat or electricity so that they accept the modified plasmid. The Agrobacterium tumefaciens is then introduced to the plant and inserts the transgene inside the plant cell's nucleus. Another method of gene insertion is using gene guns that shoot metal particles coated with DNA into plant tissue. In animals, eggs from a donor female are collected and fertilized. The gene of interest is injected into the zygote. The zygote divides many times to form an embryo whose cells all contain the transgene. A successful embryo is chosen to be placed in a surrogate and the transgenic animal is eventually born. The final step is growing the transgenic organism. As it grows, DNA replicates and cells divide. If the transgenic organism is successful, it will produce the useful protein throughout its lifetime and reproduce to pass its genes to its offspring.
Created using Powtoon.